Good evening, dear participants, dignitaries, and colleagues and resource persons. A delightful welcome for the day 5, session 9 and 10. Let us start the session 9 by welcoming our expert, Dr. Samir Syed. Dr. Samir Syed, introduced by Dr. T. Naveen Reddy. Naveen, sir. Is it audible? My voice is audible?
Yes, ma'am.
Dr. T. Naveen Reddy, kindly introduce Dr. Samir Syed for the today's session. He is there in the online?
Good evening, ma'am.
Yes, sir. Let me introduce, sir.
Yeah.
It is my pleasure to introduce Dr. Samir Syed, a distinguished microbiologist specializing in sustainable agriculture and environmental sustainability. He is currently serving as a research associate at the Northwest Institute of Eco, Environment and Resources, Chinese Academy of Sciences, Lanzhou, China. Dr. Syed received his PhD in microbiology from Sri Venkateswara University, India in 2016. After completing his MSc in 2009 and BSc in 2007 in microbiology and allied sciences. For the past decade, he has gained extensive research experience in both India and China, working as a research scientist at DRBILS, a postdoctoral fellow at Nanjang Normal University, and a DST Serb postdoctoral fellow at Angaru, India. His research focuses on plant growth, promoting rhizobacteria, biosmart nanofertilizers, and soil fertility, as well as soil microbe CO2 interactions and heavy metal bioremediation. He has published widely in peer-reviewed journals with significant contributions to microbial remediation, oxalate carbonate pathways, and also sustainable bioorganic fertilizers. In recognition of his scientific contributions, Dr. Syed was awarded the prestigious DST Serb National Postdoctoral Fellowship by the Government of India. Alongside his research, he has taught as an academic consultant at Sri Venkateswara University and contributes actively to the scientific community as a member of professional societies and as a reviewer for reputed international journals, including Process Biochemistry, Materials Letters, and the Journal of Stored Products Research. Please join me in welcoming Dr. Samir Syed. Welcome, sir.
Yeah, thank you, ma'am. Thank you for the introduction. So, good evening, all. So, it's my pleasure to be back in the session again. So, this session I'll be taking you through some of the fungal strains that are used in bioremediation and degradation of microplastics. So, let me share the screen. Is my screen visible, ma'am?
Yeah, visible,
Yeah.
sir, visible.
Yeah, thank you. So I'll be taking you through the role of fungal strains in bioremediation strategies, especially for degradation of microplastics in wastewater, and what's the future course of action that we could follow to reinforce these systems. So coming back, I mean, going through the introduction again. So maybe you'd have heard about these introductions through various presentations, but nonetheless, I'll just uh, make it too fast in these areas. So the any particle size, plastic size that plastic particle size that is of lesser than 5 millimeter in size, we term it as microplastics. So the major problem with microplastics is one is their role as such, but more than their particle size and all, these microplastics, once they become lesser than the size of 5 millimeter, they obtain properties where they could adhere lot of toxic polycyclic aromatic hydrocarbons, which actually increases the toxicity manifold. And because of their physical and chemical resilience for degradation, so these microplastics pose a tremendous risk for both biological and abiotic environmental systems. So coming to the global distribution of microplastics, so post 2014, there is increased search of microplastics that is distributed in all the environmental areas. You could see increased distribution in aquatic systems, terrestrial environments, atmospheric systems, and also all the food chains worldwide. And coming to the human, there are also recent reports since past decade where there are increased incidence of microplastics uh, in humans, especially in blood and sputum, alveolar fluid, placenta, organs, and also gastrointestinal tracts. Coming to the physical and chemical characteristics, as I told previously, the lesser than any microplastics that is of lesser than 5 millimeter particle size, we term it as microplastics. So there are different studies, even there are also different classifications, which even I presented in the last presentation. So there are also nanoplastics where the size is of nano and micrometers actually. So, and they also exist in different shapes. Uh, they might be existing as a fibers, fragments, foams, pellets, films, each with distinct environmental behavioral patterns. And uh, so their chemical composition. So as you know that these microplastics are, to be frank, plastics as such, you can classify in two forms. One is that is having only carbon-carbon bonds, and there are hetero carbon chain uh, polymer plastics. So based on their chemical composition, so it will influence the polymer type, it will influence its density, buoyancy, and surface characteristics. So that will influence what all they bind to and how much toxic it can be. Coming to the common polymer types here, so you could see polyethylene, the most abundant plastic polymer commonly found in plastic materials, exhibiting high environmental persistence. And you could also see polypropylene, polystyrene. With these three are mostly comes under, they have the whole polymer linkage is having a CC carbon chain polymers. And coming to PET and PVC, and especially polyurethane microplastics, they have which are major sources of all the PET bottles, what all the drinks, what all we are consuming. So they're all made of PET bottles, PET plastic. So they are classified under heterogeneous polymeric chains actually. So the carbon linkage will also have a carbon nitrogen carbon oxygen linkage in the polymeric chain of these plastics. And coming to the biological translocation mechanism, as I previously explained, that these microplastics are even found in the whole food chain, and also in the different areas of human body. So coming to how exactly they penetrate, so the penetration of these microplastics majorly depend upon size. So particles smaller than 10 millimeters can potentially enter circulatory systems, with those larger than 0.5 mm are less likely to pass through gastrointestinal barriers. So indicating that there is a size selective barrier, that size selective entry of these microplastics. So when you so alcoholics, let's say for example, who consume alcohol and all, you would have come across the disturbance in their gastrointestinal tight junctions. So those people might be more prone for the accumulation of microplastics. So as these microplastics entry is size dependent, so people who are vulnerable uh, for gastric intestinal disturbances in their lining, so you could see more microplastic damage to their health and also the accumulation. And coming to the biological translocation also is influenced by the microplastic chemical structure. So the chemical structure again influences what all molecules they can bind to that are highly toxic. So the surface properties also impact biological translocation. So because once they bind or adhere to the different ionic molecules, so it will change the polarity of the molecule. So they might be hydrophilic, hydrophobic, and also cationic, anionic. So these all properties determine how exactly they can be penetrated in these cellular systems. So the chemical composition determines critical traits, including the particle shape, density, and buoyancy, which influence subsurface distribution patterns in oceanic environments and biological uptake mechanisms. And coming to the microplastic pollution, the major uh, pollution ecosystem is from the land, because so this obviously says that it is a human-made. Ideally, it's originated from humans, no other. So ocean-based pollution just contributes to almost 10%, and that is also through activities, offshore activities, coastal tourism, and some of the effluents that are released into ocean. So land-based microplastic pollution is a major contributor. So are humans. And coming to the primary sources of land-based uh, microplastic pollution, so you could see that the consumer products, what all we use day by day, and even the COVID pandemic, the consumables that went for the protection during COVID pandemic, there are major sources for microplastics, and also the personal care items. So most of the cosmetics and gels, what all we use for facial creams and all, so they have micro beads. So uh, that will help in better distribution of these things and also emulsification. So most of these personal care items also use a micro bead plastics. So they also are one of the primary contributors of these microplastics. And then comes textiles, industrial activities, which actually causes primary microplastics. So the industrial materials, which includes construction materials, textiles, and industrial activities, contributed to manufacturing process and material degradation. And that transportation is another major factor, where the wear and tear of the rubber and rubber materials, either from the wheels or any automotive aesthetics. So there are also primary contributors. And coming to the ocean-based microplastic accumulation, the major sources, though ocean-based microplastics account for 10% of the microplastics in the whole environment, the major reasons for accumulation of microplastics in ocean comes from three areas. One is coastal tourism, where the seaside tourism generates plastic waste that enters marine systems through inadequate waste management practices, and of course offshore petrochemical industries and offshore industrial activities, where the plastic particles directly come into maritime environments, and also maritime activities, let's say commercial fishing operations, uh, do contribute to microplastics through the usage of their articles like fishing nets that are made up of nylon nets, and also monofilament lines. So that are some of the contributors for ocean pollution. And coming to the classification of microplastics, the primary microplastics, what wherever we use microplastics voluntarily for a beneficial or some other gain, so we demarcate that as a microplastics. Let's say the microbeads, what so ideally we make these plastics so intentionally. So these are the plastics that are used in personal care products like cosmetics, gels, and all, and also textile fibers, so which actually are helpful for the reinforcing of the cotton fabric. So there are also contributors of the primary microplastics. And coming to the secondary microplastics, these are the ones which are not made intentionally, but they are the ones which will be uh, formed in the process of primary plastic making. So, or there might be formed through degradation also. So, formed through degradation of larger plastic material, where physical weathering, chemical breakdown, or even biological decomposition. So, there are there might be a different classification based on whether they're terrestrial, marine, or so. And coming to the morphological classifications, you might find a microplastic in form of fibers. It can be in form of fragments, or it can be in form of pellets. So these morphological classification do influence the moieties these microplastics bind to. So do their toxicity and how effectively they can be degraded. So because the surface area do matter in the process of degradation, so the morphological classification do influence how the microplastics can be processed. And coming to the major topic of discussion for today, so the waste treatment plants, what most of the urban areas or most of the industries or industrial says most of the industrial says will have these waste treatment plants. They actually serve as a better entrapment mechanisms for microplastic collection. But the point is how we process these microplastics when they get entrapped. That is a major challenge here. So there are different. The next few slides, I'll take you through how exactly these are better trapped through waste treatment plants. How better we can degrade them further so that they can be brought back into the natural systems without any toxic effects. So in waste treatment plants, so the entry pathways, so the domestic sources include synthetic fibers from laundry operations, while industrial and stormwater sources contribute varying particle loads. And coming to the treatment waste in waste treatment plants, so waste treatment plants do remove 90% of microplastics through combined chemical and biological processes across multiple treatment stages. And most captured microplastics become entrapped in treatment sludge, necessitating careful management to prevent environmental release. So this is the area which uh, the role of biological entities play a major role, like how we exactly treat the entrapped microplastic through waste treatment plants. And so these are treatment efficiencies through uh, different stages of waste treatment plants. So you could see that The removal efficiencies range from almost 16.5 to 98% in primary treatment stages, where the advanced process like disc filtration achieve even higher removal rates. And degradation process in wastewater, once they get entrapped, the degradation will be of again three different types. Though any degradation starts, any plastic microplastic degradation starts with the mechanical processes, where the physical fragmentation through grinding, abrasion, mechanical stress will cause the particle size to decrease further. Then comes the chemical degradation, where the oxidation, hydrolysis, and chemical reaction that break the polymer bonds or modify the particle structure. So these chemical degradation expose the ends of polymer, so which are then susceptible for the biological breakdown. So as even you would have known that these two initial steps, the mechanical and chemical degradation, are must for a biological entity to act. They can't act on a at least currently we don't have evolved organism that can act on a complex, unbroken, not chemically degraded plastic structures. So they are the must for biological entities to act upon. So there are different microbial communities that form biofilms, especially fungi, that can contribute to gradual degradation through enzymatic processes. And in the waste treatment plants, so wherever the microplastics and the microbial community coexist, we call it as plastisphere. So these plastisphere have both fungi and bacteria coexisting together, forming a biofilms, and this itself contributes itself, or we can call it itself as an ecological niche. So this plastisphere has a different community composition. So it has bacteria, fungi, and even some other microorganisms that are resistant to that form resistant biofilms, and their composition do vary from area to area, and also the mode of treatment, and also the microplastics that get entrapped. And the microbial community do influence the particle interactions and can potentially inhibit treatment efficiency, affecting process like methane production, nitrogen conversion in biological treatment plants. And coming to the, as I told, that wastewater treatment plants currently are good enough to almost entrap 90% of the microplastics. But the point is, the degradation rates of these collected microplastics is too low through biological means or any other alternatives. So you could see that the polyethylene degradation, even after 800 days in soil-like conditions, is almost only 1.1% of the whole collected things. Same is with polypropylene degradation. But the wastewater treatment removal, you could see that it could almost enter 90% of the microplastics. So the current study is how exactly we can treat these entrapped microplastics better. So one way is hydrothermal liquefaction. It's an emerging technology that is capable of reducing microplastic concentration in treatment sludge through high temperature and high pressure processing. It offers a good promise for sludge management improvement. And another way is engineered microbial systems, which you might be also encountering all through these days, where there are different synthetic biology tools and also enzyme engineering tools to design an effective enzyme that has a high turnover rates, that is tolerant to higher temperatures, tolerant to organic solvents to be used in industry, and also broad spectrum catalytic activity on different plastics. So as you know that there are different plastics like PVCs, PETs, uh, polypropylenes, polyethylene. So the bacteria or the enzyme, whatever we wanted to choose for further evolution, need to be uh, evolved on these lines, that they should be industrially relevant. So that comes under engineered microbial systems. So we need to develop engineered bacteria and also specific enzymes that has potential to enhance breakdown rates. So that is another way where we can treat these microplastics. And fungi is another robust microbial systems that are almost ubiquitous in nature. So they are also known, different fungi currently are known for degradation of plastics and use plastics as their chief energy resources. So in next few slides, I'll be taking through different fungi that can be acted upon these microplastics and that are capable of degradation. Of course, these need to be further engineered, or these need to be uh, allowed to evolve further to make it as a robust, commercially viable microorganism systems. So the role of fungi in microplastic degradation represents an emerging and promising field within environmental biotechnology. Unlike traditional practice, unlike traditional physical and chemical methods, fungal degradation offers biological solutions that could revolutionize wastewater treatment approaches. So there are almost around 400 microorganisms that have been identified as capable of degrading plastics, with more than 70 fungal species in these 400 microorganisms, predominantly Ascomycetes species. So, which can serve as a robust microplastic remediation systems in aquatic environments. And you could see different fungal species here that are denoted, that are capable of plastic degradation. You could see some Aspergillus and Penicillium species, and there are also some marine and white rot fungi, and there are also some terrestrial soil fungi. And each fungal class is capable of acting on specific microplastic type. Coming to Aspergillus, they can degrade polyethylene, polyurethane, and polystyrene polymers. And coming to Penicillium species, they could degrade on low density polyethylene, polyhydroxybutyrate, and PET materials. And marine fungi, the Zelerion maritimum, uh, where much of the literature is also there, and a lot of studies are going on in modifying the enzymes and the metabolic fluxes in this organism especially, so where they can degrade polyethylene microplastics in marine environments. So, and also we have a white rot fungi that can degrade polypropylene microplastics, but it requires a UV pretreatment as a uh, major uh, primary step even before it can act. Mostly it enhances that. And you'll also have a fusarium and trichoderma species, trichoderma species in terrestrial environments that can come under soil fungi that also acts on this microplastic degradation. And coming to the Aspergillus species, so you could see each species of Aspergillus targets different different microplastics, and each species acting on specific microplastics also have a uh, different end product as such, and their mode of action is also different. Some acts through production of enzymes, some acts through modification of the surface, some acts through uh, biofilm formation, where they all together changes the surface chemistry of these microplastics, which can be acted upon further by some other microorganisms. So Aspergillus, there are different species here that can target different microplastic polymers with different rates and different modes of action of how they exactly degrade microplastics. And coming to Penicillium species, you could also see that Penicillium species degrade low density polyethylene microplastics, again through specialized enzyme systems and hyperpenetration mechanisms. And as I told that a lot of studies going on modification or engineering the enzymes from these marine fungal organism, Zalirium maritimum. So this marine fungus represents a specialized adaptation to oceanic environments, demonstrating remarkable ability to colonize and degrade polyethylene microplastics in saltwater conditions. Its unique enzymatic profile and salt tolerance makes it particularly valuable for marine plastic remediation strategies. The fungus form characteristic biofilm on plastic surfaces, utilizing specialized hydrophobins for attachment and employing marine adapted enzymes for polymer breakdown. And research indicates that significant potential for bioaugmentation applications in coastal wastewater treatment systems. So, being in the harsh marine conditions itself makes this organism and the enzymes present in this organism a prima facie a good choice for the picking these enzymes for further modification. So that's why a lot of studies are going for modification of the enzymes and how exactly the metabolism happens when they grow on these fungus and when they grow on these microplastics and all. And coming to white rot fungi, so again it acts through enzymatic systems, like it produces laccases, manganese peroxidases, lignin peroxidase enzymes that demonstrate strong oxidative capability for polymer chain modification and fragmentation processes. These white rot fungi are known to degrade polypropylene polymers, and UV pretreatment actually enhances their ability to act upon these polypropylene polymers to degrade to monomers. And there are different additional fungal species, some are terrestrial, some are aquatic, that do demonstrate abilities for microplastic degradation. And as I discussed previously, so fungi, if you can devise tools, or we need to target fungal enzymes for further uh, engineering them so that they can become a robust entities for ideal degradation of microplastics. As I told that it's only 0.1% of polypropylene or 0.4% of polyethylene that gets degraded in the entire sludge, whatever you collect. So the current natural entities are not capable of degrading microplastics at the rate that are getting produced by humans actually. So we need to evolve these fungal enzymes and fungal organisms also at par with the current usage and production of microplastics. And so some of the enzymes that are some of the case studies are here where we can choose these enzymes, or this can be a candidate enzymes for further modification to increase the microplastic degradation. So you have your cutinases, so which acts as a precision polymer cutters. So the mechanism of action is they actually hydrolyze ester bonds present in polyethylene terephthalate, the PET, and polyurethane polymers. Mostly these are again heterocarbon chain. So it's they have carbon carbon, also oxygen nitrogen in their polymer. So which are tough to degrade in natural means, but there are enzymes called cutinases which are capable to hydrolyze these ester bonds. So they are the better good sources for further making them more broad spectrum, more tolerant to temperatures and organic solvents to be a candidate enzymes for degradation of microplastics. So the organisms that produces these cutinases are Fusarium and Humicola species, uh, which secrete these enzymes as part of their natural metabolic processes for accessing carbon sources from synthetic materials. And these enzymes show particular promise for PET bottle derived microplastic treatment industrially currently. So addressing one of the most abundant polymer types in wastewater systems. And there are also lipases, esterases, which these are broad spectrum degraders. So lipases, these enzymes break down polyester polymers through hydrolytic cleavage, particularly effective against flexible packing materials and synthetic textiles. And mostly these uh, enzymes are produced by Aspergillus species, so making them valuable candidates for bioaugmentation and treatment systems. And the type of microplastics these organisms and these uh, enzymes target are mostly polyester derived microplastics. And apart from these enzymes, there are also some other enzymes which actually oxidizes the polymer chain of microplastics. Though they don't hydrolyze, but they actually make the polymer chain to be ready for the degradation by other enzymes through hydrolysis. So there are some enzymes here like lactase, manganese peroxidase, and lignin peroxidase from different fungal sources that actually oxidizes polyethylene through radical formation and chain modification. And manganese peroxidase do create polymer surface modifications, thereby enhancing further degradation. And lignin peroxidases, it has a high oxidation potential for most of the resistant polymer structures. So these oxidative enzymes create reactive oxygen species that modify polymer surfaces, generating functional groups that makes these plastics more susceptible for further enzymatic and microbial attack. And there is another class of enzymes which might be a good target for further enzyme engineering. Those are all depolymerases. So penicillin species produces these depolymerases, which are specialized biodegradable processors. So they act mostly on PHBs, which are very toxic, polyhydroxybutyrates, and related biodegradable polymers. These enzymes represent the cutting edge sustainable plastic processing, offering pathways for complete mineralization of biodegradable plastics. So the high specificity of these enzymes make them particularly valuable for processing biodegradable plastic alternatives, supporting circular economy approaches to plastic waste management in wastewater treatment systems. So this is the ideal way or ideal mechanism of how a fungal enzymes or fungal consortia as such acts on degradation of microplastics. So it starts from biodegradation, where the microbe attach through hydrophobins and biofilm formation on these polymer surfaces. Then they degrade these polymer surfaces by enzymatic ways into smaller oligomers and monomers. Then these monomers will be uptaken by cellular entities, either as a carbon source or any other metabolic source. And this metabolic processing then takes care of degradation. And then comes the fourth stage, where the monomers, once they get assimilated into the cells, will be converted into carbon dioxide and water, and even more smaller, simpler entities that are released into environment. And coming to the first stage, so where it's called the foundation phase, where the fungi produces specialized hydrophobin proteins that facilitate initial attachment to hydrophobic plastic surfaces, overcoming the natural repellency between aqueous environments and plastic materials. So once a fungal microbe attaches to the um, microplastic surface through these hydrophobin proteins, then they colonizes to form a biofilm, which is more robust in terms of acting together, in terms of resistance for any external entities to act on. And it also gives them a competitive advantage, like they can't allow any other organism to enter, so which is through competitive inhibition. So that is that is advantages. Once they colonize and produce biofilm, they become even more robust in their activity. So following the attachment, the fungal establishes complex biofilms that create localized microenvironments with altered pH, oxygen levels, and enzyme concentrations that enhances degradation process. The biofilm matrix provides protection for the fungal cells while concentrating degradative enzymes at polymer interface. So these biofilms do create optimal conditions for subsequent degradation. And then comes the second stage. Once the microorganism colonizes to form a biofilm, now they are set for the biofragmentation of these microplastics, which is a critical step ideally. So the biofragmentation happens by uh, first chemical modification of the ends so that the polymer get exposed. So the enzymes then different enzymes do act on ends to for a chemical that will modify the ends. Then there are enzymes that hydrolyze these polymer chains into monomers, and then these polymer chains will be formed into oligomers and monomers, which will be serve as a carbon source for the next stage for cellular uptake and metabolism. So the third stage is assimilation. So the monomer entities of microplastics now, monomeric entities from the biofragmented approach in the last stage. These
The
simpler
slide is
entities.
empty, sir.
Is it for everyone, ma'am, or it's
It is okay now.
okay?
Maybe size of the slide is.
Okay.
Yeah, sir. It is okay, sir. Now you can continue.
So this is the third stage where the cells will take up the monomeric entities for further processing. So some use them as a carbon source. So these monomeric entities will be transported through membrane, and the specialized transport proteins that facilitate uptake of these plastic-derived oligomers and monomers across the fungal membranes for intracellular processing. And these monomers then get integrated into metabolism of the organism, so that get degraded and detoxified to be taken up by the next stage. So the final stage is mineralization. So where the polymer fragments that were broken, defragmented into monomers, taken up by cells, now goes up into again environment through simpler molecules like carbon dioxide and water, or biomass, cellular biomass, if it is used as a carbon source. So the final stage achieves complete conversion of plastic derived carbon into harmless end products like carbon dioxide, water, and increased microbial biomass. This represents the ultimate goal of biological plastic remediation, complete removal of synthetic polymers from environmental systems. And this is again a repetition of previous slides where you could see microbial succession in wastewater. So as I told that initially there is a uh, microbial entity attachment, then there is a biofilm formation, then comes a community maturation where they act aggressively on microplastics to degrade. So it's not only there are different factors that affect this whole pathway. Either from attachment to release of CO2 also. So even though fungi is robust, but there are some environmental factors that affects this mechanism. So there are some temperature effects, pH, nutrient availability, and oxygen requirements that actually alter how effectively these fungi can act on these microplastics. So these need to be addressed by making fungal enzymes and fungi as such to be more robust through different evolutionary strategies, either through rational engineering or directed evolution or strain engineering. So there are different approaches where we can overcome these limiting factors that affect fungal growth and fungal activities. So to make the process even more faster, apart from fungal modifications, fungal enzyme modifications, we can also do some pre-treatments at the wastewater treatment plants that will enhance the degradation of these microplastics. Some of the processes are UV pre-treatment, which are known for most of the enzymatic activity. So the UV radiation creates a carbonyl groups and surface modifications that enhances enzyme accessibility and binding sites. And we can also do thermal processing, where the controlled heating alters polymers, crystalline and surface properties that improves the fungal colonization potential. And we can also do chemical modification of the ends of these polymers, which makes enzymes to be acted with higher turnover rates for degradation of these microplastics. And there is there is also need for selectivity of these fungals. So as I told that not all waste water treatment plants accumulate similar microplastics, and depends upon geography, depends upon the type of technology they're using. So accordingly we should uh, colonize selective fungal strains in these wastewater treatment plants as per the microplastics that are accumulated in a uh, uh, higher number. So there are some polyethylene specialists, some fungi which act specially on PE, like Aspergillus penicillium. So we could choose those organisms to degrade wastewater treatment plants that are accumulating these selected plastics. Same with PET degraders. So there are some cutinase enzyme producing organisms like Fusarium and Humicola. So we need to colonize those organisms in those wastewater treatment plants for effective removal and degradation of microplastics. Same with polystyrene and some other biodegradable plastics. Uh, and coming to the future course of action, like what all we can do to reinforce these fungal strains and the fungal mechanisms. One is, yeah, we studied about the pretreatment strategies like UV, thermal, and chemical pretreatments that makes colonization of fungi and the activity of their enzymes to be more better. It's coming to some other activities. So one is enzyme engineering approach, which we were discussing now and then. So enzyme engineering approach starts from modification of amino acids. So for the modification of amino acids, you need to modify mRNA. To modify mRNA, you need to modify DNA. So it all dwells down to modification of your genetic material of the organism that produces this enzyme of interest. So then comes once the, the codon is chosen to be modified. So uh, then there are different variations where we can target. So as I told that for a enzyme to be commercially viable or to be effective, it need to pass different tests. One is it need to be it should have a broad spectrum catalytic activity. It should act on different microplastic polymers like PET, like PU, like PS, like PP. That is one area. And second thing, it should have a higher thermal stability, higher organic solvent stability, and higher salt tolerance, or even tolerant to any other mm, ions that are present mostly in these salt-rich environments also. So the protein engineering strategies depend upon what is the target microplastic and the target environment you are working on. So this all comes under the larger domain called synthetic biology. Synthetic biology covers both enzyme engineering and strain engineering approaches. And there is also a way where we can augment these wastewater treatment plants. So there are different stages where these microplastics can be entrapped, and the effect of fungi can be augmented for the better degradation of microplastics. So there are so there can be a primary clarifiers which where the fungi of interest can be introduced uh, to target coarse plastics. Then comes aeration tanks where the bioaugmentation promotes microbial degradation under oxygenated conditions. Then comes the secondary treatment where fungi enhance breakdown and fragment to microplastic reduction. Then comes the effluent polishing with the final degradation and capture of residual particles before discharge happens. So the strategic introduction of plastic degrading fungi into existing treatment systems offer a practical pathway for implementation without requiring complete infrastructure overhaul, making the technology economically viable for widespread adaptation. And coming to the micro remediation, so it can serve as a sustainable solution. So the micro remediation, where the fungi is used for bioremediation of microplastics, can actually as a multi-pronged beneficial effects. One is it helps in ecosystem integration, where the fungal system provides multiple environmental benefits apart from
అన్నం కూడా
fungal,
హైదరాబాద్ లో ముట్టించేద్దాం ఉన్నాను ఎక్కడో చోట ఇంట్లోనే.
like soil improvement and habitat restoration and biodiversity enhancement alongside plastic remediation. And then comes carbon sequestration. Fungal biomass production during degradation processes contribute to carbon sequestration, providing climate change mitigation benefits. And also micro remediation do help in energy recovery potential for biogas production from fungal processes, creating energy positive treatment systems that offset operational costs. And another approach is omics. Like there are different organisms that are lost in the evolution. So, but these omics approaches like metagenomics applications, we can use the homology modeling simulations to choose organism or candidate gene of our choice from the extinct fungal organisms and bring back them to use for degradation of these microplastics. It's another rational approach where you follow structure based homology modeling in choosing these organisms these enzymes where current structures almost it should resemble the current plastic degradation structures. That is one way where we choose these DNA sequences from the extinct fungal species. So that is one area where we can dig deep to fish out candidate gene of our choice for a better robust enzyme to make. So another is proteomics approach, where protein analysis identifies novel enzymes and metabolic pathways involved in plastic degradation, providing targets for enhancement and optimization efforts. And another area for future robust development of these platform is we should find a species that can coexist together. So where bacteria which play major role in cycling of natural elements. So if they can coexist with fungal specialists which are actually doing the job along with algal components that provide carbon sources. So such microbial consortia, if you could identify the different bacteria and algal components that can go in sync with these fungal specialists, it's a good approach to go ahead with, where we can colonize these microbial consortia into wastewater treatment plants for effective degradation of microplastics. And another area is like, okay, we identify different bacteria, we identify different fungi, we identify different algae, but how you scale it? So the major idea comes from bioreactor design. So these are not a simple organisms like precision fermentation where we use to scale up E. coli. So these are specialized fungi and algae species, so which need a specialized bioreactor designs and all. So either you can follow a batch system or continuous flow system or even hybrid approaches to scale these organisms, so that unless and until you scale these organisms in a nutrient in a independently, so most of these organisms we don't know how exactly we can culture them in laboratory. So we need to master even that to scale them actually. Then only we can colonize these in these for degradation of plastics. And we should also address these rate limiting steps uh, by pre-treatment, like UV pre-treatment, which we discussed further. So all together, so we can address the current limitations of degrading microplastics, either by UV treatment, then comes enzyme engineering or consortium development, or even devising optimal conditions for growth of these organisms and colonizing them. So all together we can make this fungal platform a better sustainable bioremediation strategy for microplastics. So it has a much global impact and uh, for a cleaner planet. So from this, I would like to end my presentation. So I would be taking any questions if you have any. Yeah, thank you.
Dear participants, any questions? Yeah. Thank you, sir, for such a wonderful presentation and spending your valuable time.
Yeah, thank you, ma'am.
On behalf of the organizing committee, Ms. Sailaja will extend vote of thanks.
Good evening all. I extend sincere thanks to Dr. Shamir Syed for presenting novel fungal remediation strategies for wastewater microplastics. Your futuristic approach will surely guide new innovations. I am also thank our participants for their active involvement. Thank you.
So, thank you, sir. Thank you.
Thank you, ma'am.
So,
Excuse me, sir.
dear participants,
Good evening.
sir, are you there?
Yes, yes, I'm there.
Yes, sir. Somebody maybe asking.
Uh, Good evening, sir. This is Suman Sani. Can I
Mhm.
proceed?
Yes, yes.
Sir, I have one doubt. I would like to know, as you have explained, that in biological degradation,
Mhm.
microbial communities are there which form biofilms, right?
Mm-hmm. Yes.
So, can you please a little bit explain microbial communities?
See, when you say biofilm, it's not a single organism that form a biofilm. There are such organisms also, but when we are saying about this approach, it's always good to have a multiple organisms together that forms a biofilm uh, that can where each exchange different signaling factors exist together, exchange of resources like algae will give a carbon source, bacteria will give a nitrogen source, fungi do a degradation activity. So I mean a biofilm of ideal consortia. Yeah.
Okay, sir. Thank you so much.
Yes, sir. Thank you very much, sir.
Yeah. Thank you.
Sir,
Anybody?
hello.
Yeah. Tell me.
Yeah.
Sir, how these organisms affect the human?
That is another study, ma'am. Okay, so currently, first we need to identify. It's not about organisms. If any organism is known to degrade the microplastics in a better way, we can exclude that organism, but take out the enzyme of interest and scale that in bioreactors and use that enzyme. So how these organisms affect, that is second study, but the primary importance is which organism is effective in degrading these microplastics. If it's organism toxic to humans, we can remove that organisms but take the enzymes from that organisms and use it. So I don't think so that will be a major priority when we are working further.
Okay, sir. Thank you.
What type of tests used for this, like bioreactors?
Come again, sir. I couldn't hear you.
So, bioreactors used.
Sir, your voice is not clear. It is breaking.
Hello, sir. Hello, sir.
Yeah. Come again, sir. Yeah, tell me.
Sir, what type of bioreactors used for this process?
What type of bioreactors you use for these processes? So it depends
Yes, sir.
upon
Yes.
microorganisms you want to colonize. If you want to colonize bacteria, we need to use precision bioreactors. Okay, mostly these are batch based. But if we are using fungi scale up, we use continuous fermenters, where we take out the biomass, introduce the fresh nutrients, but the action goes on. Okay, because the initial seed takes a lot of time in fungi. It depends upon which microorganisms you wanted to grow. So there are different
Okay, okay.
approaches currently. Yeah.
Okay. Based on type of microorganism, we are selecting type
Yes, the
bioreactor.
fungi. Yeah, we are selecting the bioreactor.
Okay, right.
Good evening, sir. Shall I ask one question?
Yes, yeah, please.
Sir, during microbial degradation of microplastic, um,
Mm-hmm.
uh, it's like collecting samples. It's like from doing microbial degradation from collecting samples from water resources. Is it good study, or it's like uh, purchasing any microbial from other strains and isolating it for microbial degradation, which will make the good study?
So you wanted to purchase some. Okay, so idea. Okay, so the objective is you wanted the organism which can degrade microplastics. I think that is the objective. Yes. If that is the
The
objective, better.
objective is microbial degradation is the objective. My question
Yeah.
is isolating microbes from the any aquatic, any polluted resources and doing degradation makes good study, or it's like
Studies.
any known organism purchasing, or it's like
Yeah,
directly.
that is always better, ma'am. Better to go with known organism that degrade microplastics. You better do a strain engineering in the sense like evolve the strain. You evolve the strain with usage of different microplastics. That is a different step altogether. It's not that always you find a microplastic degrading enzyme from the ecological niche of microplastics. That is rare, but if you have an organism which is already known, it's better you evolve that further so that they can degrade fast. You create a different conditions like temperature, pH, and all, so that they can secrete more enzymes. So that is a better approach, I feel.
Okay, thank you.
Dear participants, any other? So, okay, sir. Now,
Yeah, thank you, ma'am.
email you. Thank you, sir. Thank you very much.
Thank you much.
Yes, sir. Dr. B. Viswanath, sir. Are you ready? Or let us take a break.
Hello.
Sir, are you ready, sir?
Of course, I'm ready, ma'am. Is it okay for 5 minutes break?
Okay, sir. Yeah.
Okay, ma'am. Fine.
So, dear participants, let us take a 5 minutes break. So, another session will start.
Hello, madam. Hello,
Sir,
madam.
tell me, sir.
Is
Tell me,
there
sir.
any exam for tomorrow, madam?
Yeah, sir. Tomorrow, same time, sir.
Exam.
Exam also, after session completion, I will send the link, sir.
Right,
I will
madam.
share the link. Yes, sir. Exam is
Saraswati
there
ma'am,
and yeah, tell me, ma'am.
good evening, ma'am.
Good
Ma'am,
evening.
you will be sharing the Google Form quiz or it will be conducted on ATAL portal. Regarding that only,
Actually,
I WhatsApp you.
yeah, you asked. Even
Yes,
both are ready, ma'am. Based on the time
okay,
and
ma'am.
all, we will share, ma'am.
Sure.
Which one is
Actually,
comfortable for you?
yeah,
We will share the same thing.
from ATAL portal, na, it gets chaotic because most of the people right now also 126 people are there now. So when we
No
all
problem. We are
do the
here
quiz now,
to solve it, everything.
sure, sir. Sure, ma'am. Sure, ma'am.
Yeah,
Thank you.
don't worry, ma'am. Yeah. Mm. So after completion of the session, you need to perform the exam. Thereafter, you need to fill the feedback form, which is shared a link from the ATAL portal. So after feedback, you will get the certificate. This is the procedure.
Saraswati ma'am, good evening.
Good evening,
Ma'am, I am
Satya.
Dr. K. Venkatalakshmi.
Ma'am, tell me.
Ma'am, good evening.
Good
Ma'am,
evening,
my
ma'am.
doubt is now you said no, there will be an exam tomorrow.
Yeah.
Ma'am, do we need to condition is there, madam? Is there any condition if we pass it, we will get the certificate
Yeah,
like that?
actually,
Any
80%
condition?
required. 80% required to get the certificate, madam. 80% pass marks required.
80%, okay, you should pass it, you should get in the exam.
Yeah. We should get in
Okay,
the
ma'am. Thank
exam
you, ma'am.
80%. Yeah, thank you.
Exam 80%.
Yeah. So
Okay,
feedback
ma'am. Okay, ma'am.
thereafter
Thank you.
there is a feedback form from the ATAL
Okay.
portal. So you need
Okay.
to fill regarding all the sessions.
Okay,
So
ma'am.
thereafter automatically it will generate certificate.
Okay.
Yeah.
Okay, ma'am. Thank you, ma'am.
Okay. Yeah, thank you.
కెమెరా డోర్ వేసుకున్నాను.
Saraswati ma'am, are you there?
Yes, ma'am. Yes, ma'am.
Ma'am, I have some doubts. Ma'am, this exam will be there, no? Is it MCQs, madam? MCQs, and can you tell me?
MCQs only.
MCQs.
MCQs, 20 questions.
20 questions MCQs.
Yeah.
Okay. And madam, tomorrow's schedule and everything, will be posted in the group, madam?
Same time, ma'am. Evening
Same
only.
time. Same time. Okay, ma'am. Thank you.
Yeah, thank you. So, good evening once again, dear participants. Let us start the day 5, session 10. A heartily welcome to Dr. B. Viswanath sir, who is one of my advisor and for his constant, continuous support, and also he played a pivotal role in the success of FDP. So thank you, sir, once again. Now I invite Miss D. Manjusri to introduce Dr. B. Viswanath, sir.
Good evening, madam. Am I audible?
Good evening. Yeah, audible.
Okay.
Ma'am, excuse me. Ma'am,
Yeah.
you are audible, but if you don't mind, could you please increase the volume, madam?
Okay,
Volume.
okay, ma'am.
Okay, ma'am. Okay, ma'am.
Okay.
Yeah.
Okay,
Okay,
ma'am.
Your
ma'am.
voice is good. Maybe my voice is low, I think so.
Madam, is it okay? Should I continue?
Yeah, yeah.
Ma'am,
Ah,
little bit more, madam, if possible, please.
little bit more. Okay, ma'am. I will try. I will try.
Okay,
Now
ma'am.
is it okay, ma'am?
No,
It's
madam.
audible. Ma'am,
It's audible, but
For
very
us,
low, madam. Very low.
it is good, ma'am.
Okay,
Very,
madam.
yeah.
Okay, madam.
Maybe
ma'am, please
you
check
can
your
continue.
connection.
Oh,
Yeah,
okay,
okay,
ma'am.
ma'am.
Okay.
Let us
Okay.
continue.
Yeah,
Okay,
ma'am.
ma'am.
I'll resign, madam. Okay, ma'am.
Okay, ma'am. Okay. Okay, ma'am. Thank you, Saraswati, madam, for giving this opportunity to introduce Dr. B. Vishwanath, sir. Dr. B. Vishwanath, sir, founder and director, Dr. B. Buddolla Institute of Life Sciences, Tirupati. Ladies and gentlemen, distinguished guests, and enthusiastic participants, it's my pleasure to introduce to you our speaker for both session 10 and session 13 of the ATAL Faculty Development Program on microplastics as emerging pollutants, analytical and monitoring perspectives. Please join me in welcoming an eminent researcher in environmental microbiology, Dr. Buddolla Vishwanath, founder and director of Dr. Buddolla's Institute of Life Sciences, Tirupati. Dr. Vishwanath is a scientist whose career beautifully combines an academic excellence with social responsibility. With a PhD in microbiology from Sri Krishnadevaraya University, he has dedicated his research to exploring how enzymes, fungi, and microbial systems can detoxify the environment, addressing pollutants ranging from xenobiotics to pressing problems of microplastics. His pioneering work on Stearium australia and its lactase enzyme has opened promising pathways for eco-friendly bioremediation. With over 50 publications, 12 books, 4,000 citations, and an H index of 30, Dr. Vishwanath has made significant contributions to the field of environment research. He currently serves as chairperson of the Institutional Animal Ethics Committee and continues to advance research in areas such as microbial imaging, chromium detoxification, wastewater treatment technologies, and probiotic-based cleaning products as sustainable alternatives for environmental cleanliness. Ladies and gentlemen, let us warmly welcome Dr. Buddolla Vishwanath garu. Welcome, sir. Hello.
Yeah, ma'am. Wait, I think he will come. He will join with us.
Okay, ma'am. Okay, ma'am.
Yes, yes, ma'am. Yeah. Thank you very much, ma'am. Hello.
Hello. Oh,
Yes, sir.
Hello.
Audible.
yes, sir. You are
Audible,
audible.
sir.
You are audible, sir.
Thank you,
Welcome, sir.
ma'am.
Welcome. Welcome, sir.
Yeah, thanks to the organizing committee for inviting me for this program. I don't know how I'm going to satisfy because, of course, it's a continuous lectures. A similarity is there with the bioremediation and all those things. So, little bit I'll make change in this one. So, can I share my class here, please?
Yes, sir. Yes.
Just a second. Yeah, my PPT is visible there.
Yes, sir. It is shared and visible.
Yes, sir, your PPT is visual to us.
Yeah, good evening, all the participants. Yeah, every day we treat millions of liters of wastewater, but hidden inside are invisible enemies, microplastic, smaller than 5 mm, escaping silently into our rivers, soils, and even our bodies. They are not just environmental debris, there are biological time bombs linked to endocrine disruption, oxidative stress, and long-term ecological collapse. Let me ask you, if asbestos was the silent killer of the 20th century, are microplastics the asbestos of the century? Conventional wastewater treatment plants can remove large pollutants, but microplastics pass through undetected. They accumulate in fish, in the food chain, and ultimately in us also. We are already drinking plastic, breathing plastic, and eating plastic, coating food. The question is, how long will we wait before acting? This is where science meets sustainability. Imagine a system where membranes act as sieves to trap microplastic while living biofilms. Nature's microbial factories attack, degrade, and dismantle them into harmless compounds. This is the vision of integrated membrane biofilm hybrid system. Not just filtered plastics, but actually destroying them at the molecular level. As a faculty, you hold the power to transform this challenge into an opportunity. PhD project, interdisciplinary collaborations, and translation research that can make wastewater plants into plastic degrading factories. What we discussed today is not just science, it is a call for innovation, collaboration, and leadership in solving one of the humanity's most urgent problems. Let me take you into the invisible world where we often ignore. Microplastics are tiny, but they are everywhere in the clothes we wear, the cosmetics we use, the packaging we throw away. Primary ones are made intentionally, like beads in toothpaste or face scrubs. Secondary ones are born from the slow death of plastic bottles, bags, crumbling into fragments over decades. The problem is these fragments do not respect borders. They travel through drains, sneak into wastewater and escape most of our treatment systems. Every day, they slip silently into rivers and oceans, carrying not just themselves, but toxic additives and observed pollutants. We believe we are cleaning water, but are we really? What looks crystal clear may still carry thousands of plastic particles, invisible to the naked eye, but powerful enough to disturb hormones, trigger inflammation, and bioaccumulation in ecosystem. So here is the challenge. We can't see them, and conventional treatment can't stop them. How do we fight them? That's the journey we are about to explore today. Yeah, when it comes to the why microplastics are the matter, microplastics are not just tiny fragments. They are chemical cocktails in disguise. Their surface absorbs toxic heavy metals, pesticides, or persistent organic pollutants. When they enter living system, they act like Trojan horses, releases these toxins slowly. From a biological perspective, they disturb cellular homeostasis, triggering oxidative stress, interfering with mitochondria, and disturbing endocrine signaling pathways. Imagine synthetic plastic molecules mimicking natural hormones, binding to estrogen or androgen receptors, altering developmental and reproductive health. From a chemistry angle, the additives in plastics, phthalates, bisphenols, frame, restorers, leach out under the heat and UV exposure. These are xenobiotic compounds foreign to the body that intimate inflammatory cascades. So microplastics are not inert debris. They're active biological time bombs. And here is a big question. If asbestos silently destroyed lives in the 20th century, will microplastic will be the silent killers of our century unless we act faster? So there are conventional methods. So what are the limitations of conventional waste treatment? Our wastewater treatment plants are designed for organic matter, nutrients, and pathogens. Primary sedimentation removes large particles. Secondary treatment biodegrades organic pollutants, whereas tertiary polishing the clear nutrients. But microplastics laugh at this system. Why? They are too small for sedimentation. They are too chemically stable for microbial breakdown. Their hydrophobicity lets them bypass conventional filters. Even advanced coagulation, fluctuations, and chlorination fail to degrade polymers like polyethylene, polystyrene, or PET. The result, up to 90% of microplastic escape untreated into the environment. This is the core chemical biological mismatch. We are using 21st century method to fight a 21st century pollutant. The truth is, separation alone is not enough. Degradation is essential. So when it comes to the why it is required, hybrid systems. So complex pollutants demand complex solutions. A single line of defense won't work. This is where the hybrid concept emerges, coupling membrane separation with a biofilm-driven biodegradation. Think of a membrane as a chemical gatekeeper, sieving, rejecting particles at nanometer scale. Now add biofilms, which are microbial consortia that act as biology's living factories, secreting enzymes capable of breaking tough polymers into monomers. Together, they form synergistic shield. Capture plus catalysis, filtration plus fragmentation, separation plus mineralization. In essence, it's engineering plus ecology. This hybrid thinking is the future of water treatment, not just containing plastics, but converting them into harmless end products like carbon dioxide, water, and microbial biomass. That's why today's session is about inspiration through integration, uniting chemistry with biology for sustainable solutions. When it comes to the membrane technologies for the microplastic removal, membranes are the chemical sieves of modern science. Ultrafiltration, nanofiltration, reverse osmosis, each function on the principle of size exclusion and selective permeability. From a chemistry standpoint, membranes exploit polymer chemistry, pore size, hydrophobicity, and surface charge determine what passes and what stay behind. They trap microplastics by preventing particles larger than their molecular cutoff. But here lies the paradox. While they filter efficiently, they also suffer fouling organic matter, salts, and even biofilms clog the pores, reducing efficiency. Cleaning requires chemicals which add cost and secondary pollution. So while membranes act as barriers, they cannot destroy the microplastic. They are half the solution. And that's why integration with the biology become essential. Enter the world of biofilms, microbial communities that attach to the surface and build complex extracellular polymeric matrices. Within this slimy matrix, microbes share nutrients, exchange genes, and secrete powerful enzymes. From a biological lens, biofilms are cooperative ecosystems. Pseudomonas, bacillus, and certain fungi secrete esterases, lactases, cutinases, and petases that break long polymer chains into smaller oligomers and monomers. From chemistry perspective, these enzymes attack bonds. Esterases, as you know, hydrolysis, ester linkages in PET. Lactases oxidize aromatic rings in polystyrene. Cutinases target polyester coatings. Biofilms, therefore, are not waste. They are living reactors. Imagine thousands of microbial factories working simultaneously, each producing chemical tools to dismantle plastic. This is nature power. We just need to harness it. When it comes to the concept of membrane and the biofilm hybrid system, now imagine merging the strength of chemistry and biology into one system. The membrane acts like a net, capturing microplastic that would otherwise escape. The biofilm colonizes the membrane surface, secreting enzymes that break down trapped particles. This is not just filtration, it is filtration plus transformation. The hybrid system converts pollutants into biomass, carbon dioxide and water. It's sustainable, self-generating, and energy efficient. We are not just filtering plastic, we are engineering nature and technology together to destroy them. This is the kind of system future wastewater turns need: intelligent, adaptive, and truly circular. When it comes to the mechanism of action, let's break it down scientifically. The membrane holds back microplastic through size exclusion. Biofilm microbes release oxidative and hydrolytic enzymes, cutting bonds, polystyrene, PET, and polystyrene. The fragments become monomers like terephthalic acid, ethylene glycol, which microbes further metabolize. The final products, carbon dioxide, water, and biomass, integrate back into the natural cycle. This is a polymer chemistry meeting microbial metabolism. What plastics resist in decades in nature weathering, biofilm membrane system can accelerate in weeks. It's the dream of green chemistry, turning pollutions into harmless molecules by mimicking nature's own pathway, but faster. So, bench. When it comes to the bench scale studies and the lab scale studies, at the bench scale, researchers are already proving that hybrid systems work. Experiments show 90% of removal of microplastic when membranes and biofilms are coupled. Because the membranes trap the plastic fragments while the biofilm break them enzymatically. In control studies, plastics that would otherwise persist in centuries are converted into biomass and carbon dioxide within weeks. When it comes to the chemistry perspective, we see the reduction of polymer crystallinity, hydrophobicity, and molecular weight. From the biological perspective, microbes adapt to use microplastic-derived monomers as their food. The message here is powerful. Science is always turning into impossible pollutant into a manageable resource. If this can be achieved on the bench, imagine what happens when scaled to wastewater plant. When it comes to the pilot scale application, science is not stuck in the lab. Pilot plants are now testing these systems in municipal wastewater treatment. What do we see? Now only higher microplastic removal, but also lower sludge burden. Why? Because biofilms consumes trapped plastic, reducing the solid waste that would otherwise require disposal. This is the essence of sustainable chemistry and the biology working together. Fewer chemicals for treatment, lower energy demand, less sludge generation. These pilot projects show us the path forward. Hybrid systems can be scaled, practical, and economically viable. So faculty participants here should ask, why not test such pilot in our local waste treatment plants? Yeah, when it comes to advanced membrane materials, now let's go deeper into chemistry. Membranes today are no longer simple polymer sheets. Researchers are demanding functionalized membranes with hydrophilic coatings, anti-fouling layers, and nano-enabled surfaces. Graphene oxide, titanium oxide, and silver nanoparticles are being embedded in membranes. What does this do? Graphene oxide improves hydrophilicity and mechanical strength, whereas titanium oxide nanoparticles provide photocatalytic activity, breaking down polymer under light. Whereas silver prevents pathogenic biofilm overgrowth. This is materials chemistry solving biological bottlenecks. By designing surfaces at the nanoscale, we encourage beneficial microbial colonization while suppressing harmful fouling. It's a marriage polymer science, nanotechnology, and microbiology. The perfect example how interdisciplinary research can rewrite wastewater treatment. Then coming to the microbial engineering for the biofilm efficiency. Nature has given us microbes, but biotechnology allows us to make them smarter. Using synthetic biology like CRISPR, we can design microbes that act like a plastic assassin. Imagine a strain Pseudomonas engineering to induce PETase enzyme or a fungal biofilm program to secrete powerful lactase continuously. These microbes don't just survive on plastics, they thrive. From biological view, we are enhancing gene expression, pathway regulation, and enzyme secretion. From a chemical view, we are accelerating bond cleavage, oxidation, and hydrolysis that would otherwise take centuries. This is where faculty research can play a role. Genetic engineering, metabolic modeling, enzyme immobilization. In short, biology becomes programmable chemistry for sustainability. When it comes to the synergistic role of membrane surface properties, the success of hybrid systems depends on the conversation between surface chemistry and microbial biology. A rough membrane surface provides anchoring sites for biofilms. A hydrophilic surface allows better nutrient exchange and microbial colonization. Surface change influences the microbial adhesion and enzyme activity. Scanning electron microscopy images reveal how biofilms spread across membrane pores, creating a living layer of catalytic activity. This is synergy at its finest. The surface chemistry of the membrane determines how well the biology of the biofilm performs. By turning both, we create highly efficient, self-sustainable systems. Every membrane pore, every microbial colony becomes part of a living factory, dismantling plastics at the molecular level. Yeah, very important thing, the challenges and the future directions. Every innovation carries challenges. Hybrid membranes, biofilm systems are powerful, but they are not perfect. Why? Membranes clog, biofilms overgrow, and the costs rise when scaling up. But this is not a reason to stop. It is a reason to innovate further. We must integrate polymer chemistry, microbial biology, nanotechnology, and artificial intelligence to overcome these barriers. Today's challenges is tomorrow's research proposals. When it comes to the challenges in scale-up, at the laboratory bench, we can achieve more than 90% microplastic removal, but in the real world, problems arise. So membrane fouling increase maintenance cost, maybe biofilm overgrowth clogs the pores, reducing flow. Maybe the process economics demand efficiency with affordability. From a chemist perspective, fouling results from hydrophobic interactions and scaling salts. From a biological perspective, uncontrolled biofilm colonization becomes detrimental. The real task is balance. Engineer membranes with anti-fouling chemistry while directing biofilms towards beneficial enzymatic activity. This is where faculty research can provide solutions, scale up challenges, or opportunities for innovation. Yeah. When it comes to the environmental and ecological safety, every biological solution must also ask, is it safe? Could engineered biofilms release pathogens? Could degradation intermediates of plastics be more toxic than the plastic themselves? What are the long-term ecological impacts? Chemically, some breakdown fragments like bisphenol A or phthalates may disrupt hormones, or biologically unmonitored biofilms may harbor opportunistic pathogens. Therefore, risk assessment, toxicity evaluation, and eco-safety studies are essential. The goal is not just removal, but safe removal. This is translational research, bridges lab findings with societal safety. When it comes to the analytical challenges in microplastic detection, one of the biggest obstacle is seeing the invisible. Detecting and quantifying the microplastic in wastewater is complex. Techniques involved, so all the chemists knows it well, FTI spectroscopy for the polymer fingerprinting, Raman spectroscopy for the chemical identification, the pyrolysis GCMS for thermal decomposing analysis. But these are expensive, time-consuming, and lack global standardization. Imagine developing AI-powered microfluidic chips that detects and classify the plastics in real time at treatment plants. This is where the chemistry, instrumentation, and data science must join forces. When it comes to what's the role of AI in smart monitoring? This is the age of intelligent water treatment. AI and biosensors can revolutionize hybrid systems. Machine learning models predict biofilm activity and enzyme expression. Smart sensors detect plastic fragments at the nanoscale. Real-time data allows adaptive membrane cleaning cycles. Think of wastewater plants where every membrane has embedded sensors reporting its health and plastic degrading performance. This is not science fiction. It is where interdisciplinary faculty research can lead us. Chemistry and biology provides the mechanism. AI provides the intelligence. What are the emerging trends? Biofilms, membrane, photolytic hybrids. The future lies in multifunctional hybrids. Imagine coupling membranes, biofilms, and photocatalysis. Membrane captures plastics. Biofilm secretes enzymes to degrade them. Photocatalysts like TiO2 under light generate reactive oxygen species, accelerating polymer breakdown. This triple synergy, separation, biodegradation, and photocatalysis, offer faster, cleaner removal. This is chemistry powered by photons, plus biology powered by enzymes. Together, they create a sustainable future. When it comes to the translational research opportunities, the biggest challenge and the opportunity is translation. We must bridge laboratory research with industry, municipalities, and the policymaker. Textile industries can adapt hybrid systems to capture fibers. Plastic factories can recycle monomers recovered from biodegradation. Municipal boards can test pilot plants for urban wastewater. This is the faculty's role to move from papers to practice, from theory to technology, from science to social impact. Because The goal is not just innovation. The goal is implementation. Yeah, when it comes to the global perspective, microplastics are not a local issue. They are planetary challenges. The European Union plastic strategy is pushing for circular economy and advanced treatment technologies. The United Nations SDG 6 demonstrates cleaner water and sanitation. Countries like Japan, Germany, Korea are pioneering hybrid systems for wastewater innovation, including South Africa, where the Durban University of Technology, the wastewater technology, is running the same thing. And where is India? We are at the crossroads. With our scale of population, water challenges, India can lead in developing affordable, scalable biofilm membranes hybrid systems. This is not just environmental chemistry or applied microbiology. This is India's opportunity to lead globally in wastewater innovation. Yeah. Before going to this one, I want to check the vision of plastic-free water. Yeah, close your eyes and imagine rivers without floating plastic flames, oceans free of synthetic debris, aquatic life thriving again. Every membrane pore becomes a gatekeeper. Every biofilm colony becomes a factory of enzymes. Together, they transform wastewater treatment plants into plastic degrading factories. Hey, this is not an utopia. This is science waiting for us to act. The chemistry of materials and biology of microbes are already here. What we need is the will to integrate them. Yeah, now the real ignition point. What does this mean for you as a faculty? Yeah, you can make PhD topics and James engineering, membranes for the surface chemistry, AI for microbial dynamics. And the funding opportunities are there, DST, DBT, and international collaborators. As I want to reveal one thing here, Dr. Buddolla's Institute of Life Sciences has started in the COVID time, 2020 and 21. And we are now collaborating with Chinese Academy of Sciences in making soil salinization free from, and we are collaborating for this microplastic degradation enzymes using probiotics. And of course, you people are having university faculties and the IITs and the NITs and from other faculties. You all have the great opportunity than the institution like us. So microplastics are so new that the field wide open. Every project, every student thesis, every funded program can be the first of its kind of contribution. This is your chance to shape the next generation wastewater research. Don't stop with research. Bring this vision into our classrooms. Develop laboratory modules, microplastic detection with FTIR, biofilm growth assays, membrane performance tests. Inspire students by connecting microbiology nanotechnology, and polymer chemistry into one. As I told, encourage innovation clubs to design local solution for wastewater treatment. When students see faculty passionate about real world challenges, they too become innovator. This is how education transforms into impact. Yeah. Now the question, call for collaboration. No single discipline can solve the microplastic crisis. We need microbiologists to find and engineer new degraders, chemists to design advanced membranes and catalysts, environmental scientists to test real-world applications, data scientists to create smart monitoring tools. Collaboration is the real catalyst. Together, we can build networks that cross departments, universities, and countries. Let us transform the competition into collaboration and turn science into solution. Yeah. When it comes to the summary and the takeaway, let's pause and reflect. Microplastics, invisible, urgent, and the biologically dangerous. Conventional wastewater treatment, insufficient. Hybrid membranes, biofilm systems like most promising solution, filtration plus biodegradation. Challenges definitely remain. Fouling, safety, detection, and the cost. But the opportunities massive. Research, teaching, innovation, and policy. The message is clear, man. We have the science. Now we need the courage to implement it. Yeah. Here are some unexplored research projects we are planning to submit to the funding bodies. The first one is hybrid membrane biofilm systems for the textile microfiber removal. We are collaborating with Madras Silk Academy for this program, and they are funding for this program. And the second one is the CRISPR engineered microbes for the plastic degradation on membranes. Of course, we have submitted this for the DBT and we are waiting for the decision. And the circular economy integration of plastic degradation, this is one of the PhD work going on. And then the project is eco-safety assessment of biofilm membrane bioproducts. This is another PhD in the Reva University. We are collaborating with these things. And of course, as a small institution, we made a large contribution. We have published papers with wastewater technologies in the trends in analytical chemistry. And one paper is under review in the advanced sciences. And another one published already in the International Journal of Biodegradation and Biodegradation. And yeah, this is my wish to exchange with all you people. The battle against microplastic innovation is our greatest ally. Integrated membrane biofilm hybrid system turn wastewater treatment from mere filtration into a living, a self-generating solution, seamlessly removing and biodegrading pollutants at the molecular level. Yeah, finally, I would like to thank to the organizers for the great opportunity. And next minute, I may not available because as my father admitted in the cancer hospital. So will you please forgive me? And tomorrow I have a session And definitely, I'm going to clear the doubts of today and tomorrow together. Thanks for the opportunity. Thank you, ma'am. Hello.
Thank you, sir.
Hello.
Thank you for your nice session. So let me invite to introduce, let me invite to give vote of thanks, Dr. M.S. Pradeep. Pradeep, sir.
Ma'am, questions are welcome, but you please collect the questions. Definitely tomorrow I'm going to discuss all the questions.
Okay, sir.
Thank you, ma'am.
But
Thank you very much, ma'am.
yeah, thank you, sir. Thank you very much.
Yes, ma'am.
It's audible.
Yeah, audible, sir. Audible.
Okay, audible. Good evening, one and all. On behalf of the FDP committee, I thank Dr. Buddolla Viswanath, Director, Dr. Buddolla Institute of Life Sciences, Tirupati, for his engaging talking on integrated membrane-biofilm and hybrid system for microplastic removal. Your work has laid a foundation for practical applications. Thank you all for being such an attentive audience. Thank you, Nandal. Thank you. Hello, ma'am. Thank you,
Thank
ma'am.
you, Dr. M.S. Pradeep, sir, for nice talk. Yeah, thank you, sir. Viswanath, sir. Thank you. At this time also you joined with us. We don't have any other alternate, so that's why we troubled you. Any questions, just message us and we will forward to Dr. Viswanath, sir. Even I will share his mail ID. You can contact him for any other for further assistance. Dear participants, now you can message any doubts regarding this session and any questions.
కదా తీసుకొచ్చిస్తలే నీకైతే బండి కావాలి అంతే కదా ఎవడెక్కడున్నా పోనీ
Hello, madam. Hello, madam.
Sir, you may leave the session and just give the feedback.
Mad feedback posted in group or
Yeah, sir,
here?
we will post in the group, sir. No problem.
Okay, madam.
Because his father condition was very serious. That's why he left.
Okay, no, madam.
Sir. So it is an unavoidable condition. So that's why, sir. Okay, sir.